Good afternoon, everyone. My name is Natalie Mikhail, and today I'll be presenting research on how the gut microbiome influences healthy aging and how it could also predict longevity. Now, this is all based on a study done by Wolmansky and his colleagues in 2021, published in the Nature's Metabolism and titled Gut Microbiome Pattern Reflects Healthy Aging and Predicts Survival in Humans. So, to begin, why does this topic matter? Now, the gut microbiome has been increasingly recognized as a key factor in aging. With our populations aging worldwide, it's important to understand what can promote longevity and healthy aging. Now, this study specifically investigates whether gut microbiome composition can predict survival in older adults. And throughout the study, the researchers identified a distinct sort of microbiome pattern that has been linked to longevity, which suggests that certain microbial changes are actually beneficial for healthy aging. For some background that can help understanding the gut microbiome and aging, we can see that the gut microbiome consists of trillions of microorganisms, and this can include different types of bacteria, fungi, viruses, and they all play a role in digestion. They also play a role in different immune functions and your metabolic health, of course. However, as we age, our microbiome composition tends to shift, and this is normal since it's kind of influenced by factors such as one's diet, if they take any medications for any reason, and their overall health, such as exercise, lifestyle, or whatever they're genetically predisposed to. While some shifts are indeed natural, however, others can contribute to something called dysbiosis. And dysbiosis is an imbalance which is associated with inflammation and other metabolic disorders. And in an article written by Haran and McCormick in 2021, they actually found that it was also related to neurodegeneration. Now, research suggests that maintaining a diverse and resilient microbiome can actually promote longevity and reduce age-related disease risk. In this image, we can see what a healthy gut looks like in cartoon format, and we can see that it consists of many different types of microbes in there. However, when you have gut dysbiosis, it tends to become similar microbes to one another, and you have less diversity in there. This topic is actually very important to us and we should care about it. However, some of you might wonder why this actually matters. We can see that age-related diseases will continue to rise worldwide, and this can include cardiovascular disorders, neurodegeneration, and other metabolic syndromes. These all actually will increase our mortality rates, and it is not good for the overall population's health. However, we could also see that if specific microbiome patterns can predict longevity, then they can actually serve as early biomarkers for different health risks, which can help with preventative treatments and help people be diagnosed early on. Additionally, we can also see that they can be used for interventions such as probiotics and prebiotics, and different dietary changes can also be developed to promote healthy aging, and this will happen through changing or altering the gut microbiome. The research also aligns with the growing interest in personalized medicine, in which we could end up profiling an individual's microbiome and guiding individualized health strategies. Within this article, we can see that the researchers actually aim to answer the main question of how does the gut microbiome change with aging, and can it predict survival? Now, their hypothesis was that greater microbiome uniqueness and many beneficial microbial shifts actually will correlate with longevity, which can also suggest that rather than following a uniform pathway, the microbiome of an individual who has been identified as long lived can actually exhibit distinct patterns that show resilience against different age-related diseases such as cardiovascular disease and neurodegeneration. To address this specific question, the study actually used a population of 9,000 individuals that have ages ranging from 18 to 101, all across multiple different cohorts. For the techniques that they used shotgun metagenomic sequencing, and this was used to profile the microbiome composition sort of at a higher resolution. And in an article written by Pasternak, we were able to break down the shotgun sequencing process a little more to find that there are really 4 main important steps. And this is first extracting the microbe sample that you are trying to find, and then you are then going to sequence it to get your raw readings. You're going to continue to assemble it to get the assembled sequences, and then this can move on to data analysis. Now, this technique allowed the researchers to identify microbial species and their functional genes as well. To analyze the data now, they use longitudinal approaches which kind of track changes over time in order to assess the correlations between microbiome diversity and survival outcomes. They also wanted to use statistical models in order to compare statistics of longevity to composition, and they also adjusted this for factors such as diet and one's medication use to ensure the most accurate conclusions. For the results, one of the study's most striking findings actually was that the gut microbiome will continue to increasingly become unique with age in healthy individuals. In contrast, less healthy individuals will tend to exhibit microbiomes that remained relatively steady or actually lost diversity. They also found that younger individuals tended to have more similar microbiomes to one another, and this is likely due to their shared dietary habits and also environmental exposures. Along the same lines, greater microbiome uniqueness in older adults was correlated with lower levels of systemic inflammation, which shows a protective effect of their diversity in their microbiome. This supports the idea that the microbiome is adaptable and it's not a uniform environment. It's actually a marker of one's health and resilience to different systemic diseases such as cardiovascular disease and neurodegeneration. We can see here, this is the figure that shows the associations between the gut microbiome uniqueness and age. And specifically in these 2 side graphs, we can see that there is the microbiome uniqueness score on the y-axis, which is plotted against the participants' ages on the x-axis. In both of these, we can see that there's a upward and clear trend that can show that healthy aging individuals will exhibit increased microbiome uniqueness over time. Especially we can see it when one becomes at a much older age than younger healthy individuals. There is a significant change from where their microbiome started to where it ended. Now, this difference in the microbiome adaptation is actually a key determinant of longevity. Another important finding that the researchers found within this study was that specific microbes were actually enriched in individuals who were long-lived, while other microbes declined. Now, they found that there are beneficial bacteria that have increased in individuals with longer lifespans. These are known for their roles in barrier integrity and protection. They also serve as anti-inflammatories, which could be really good for individuals who are more prone to systemic diseases. And it also helps with metabolic regulation. However, on the other side, they found that pro-inflammatory bacteria, and these are bacteria that are associated with age-related diseases, actually declined in long-lived individuals. Now, we can see in this figure, which shows the gut microbiome aging patterns, which correlated with survival, and it presents the survival curves, which shows the microbiome composition. Now, in the x-axis, we can see the years, and the y-axis shows the proportion of surviving individuals. Those who tended to have a microbiome that had greater uniqueness and beneficial microbial shifts had significantly improved survival rates over time. Now, notably, the researchers found that these microbial shifts were actually independent of common factors such as one's diet and medication use. This finding actually strengthens the argument that the microbial composition of an individual can actually play a role in longevity rather than just reflecting one's external lifestyle factors such as eating healthy versus not eating healthy, exercising a lot versus not exercising a lot. In addition to the data that they found that showed taxonomic shifts, they also saw that there were functional changes in the microbiomes of long-lived individuals. They saw that there were increased genes that are actually related to butyrate production, and butyrate is a short-chain fatty acid, which is known to be anti-inflammatory, and it's actually gut protective. It has many benefits in that sort. They also saw that there were decreased genes that were associated with pathogenic potentials, which shows that there was a reduction in harmful microbial activity in those who were long-lived. Now, in this figure, we can see that it shows the reflection of the gut microbiome uniqueness in plasma metabolites, and this specifically shows a metabolic heat map over here, which can compare the plasma metabolite profiles in individuals with high versus low uniqueness. The x-axis actually will list the metabolites, while the color gradient will show the different concentration changes, and this can highlight the shifts that can contribute to enhancing your metabolic function and stability, and also reducing inflammation. We can see that the warmer colors will indicate higher concentrations of beneficial metabolites, such as those that are, again, linked to the anti-inflammatory pathways, while there are cooler colors which will show the reduced level of harmful metabolites, which are associated, again, with metabolic disorders. Now, the clustering patterns on the heat maps will suggest a distinct metabolic pathway or sort of signature in longer lived individuals. This further supports that there is a link between microbiome function and health as well. Now, this can demonstrate that individuals with more unique microbiomes exhibit very distinct metabolic profiles. So it's easy to tell apart from those who do not have unique microbiomes. Specifically, we can see that there were metabolites that were related to lipid metabolism and anti-inflammatory processes that were actually upregulated in order to support a longer-lived individual, while those that are linked to pro-inflammatory functions were downregulated to prevent the harmful microbes being in the microbiome. This will suggest that the microbiome uniqueness is not only microbially distinct, but it can also function optimally as to further promote one's longevity and healthy aging as well. So what does this all mean? These findings actually show us and provide very strong evidence that your gut microbiome is not just something that sits in the background of your bodily functions, but rather it actually is actively playing in your health and longevity as well. However, there's a few important considerations to make specifically about this study, which is that it was an observational study. And according to Boyko in 2013, observational studies means that it actually only establishes the correlation, but it doesn't necessarily show us what is the actual causation. Now, because of this, experimental studies are further needed in order to determine whether if we alter one's microbiome, can this actually extend their lifespan as well. We also need to see that factors such as diet, exercise, one's medication, and also their genetic predisposition can shape their microbiome greatly, and it can also alter their longevity. Some people are more prone to specific diseases that may not help them live long, healthy lives, while others do not have these genes that can cause that. However, these findings do support the idea that the gut microbiome composition actually can further help our healthcare and can serve as a biomarker for healthy aging, and it can actually have potential applications in preventative medicine in order to prevent harmful diseases, but also can be used for individualized medicine as well to further attack and treat patients based on their needs. To conclude, the gut microbiome actually evolves very uniquely in healthy aging individuals, which has been shown to have greater diversity and many beneficial shifts which correlate with longevity. Now, we've also seen that there are functional changes such as increased butyrate production, which actually further supports that the role that the microbiome plays in helping and promoting one's health span as well. We could also see that future research actually needs to be done, especially to focus on the interventions such as dietary modifications or even specific pre- and probiotics in order to further assess whether changing and altering the microbiome can actively promote one's health span. Based on this research and other research that I have done in my personal time, there actually were a few questions that I thought could branch from this topic and further allows me to think into it. The first being, could we actually slow down our aging by modifying our microbiome? Is it possible to slow down the process despite what genetics say? Another being, how can we actually use this research to apply it to individualized medicine? Since it's mainly all hypothetical that we could target a person by their microbiome, is it actually possible? Now, what are other external factors that can influence gut microbiome changes other than aging? We understand that the most common one is diet, but is there something deeper, such as specific diseases that aren't related to aging? Again, we can ask what challenges actually arise from using these microbiomes as a biomarker. And finally, how does one's diet and lifestyle impact the microbiome aging patterns? Is it true that if you eat healthier, you'll have healthier microbiomes and live longer? Or does it not really matter as much as we think? And that is all I have. Thank you, and I look forward to discussing it. And here are my references as well.